It's our honor and privilege to welcome Professor Rajeev Varshney, International Chair in Agriculture and Food Security, Murdoch University, Australia. Dr. Varshney has an admirable and stellar CV, and I will be reading out some of the major highlights.
You don't need to read here.
Professor Varshney is an agricultural research scientist specializing in genomics and molecular breeding with over 20 years of service in developing countries in sub-Saharan Africa and Asia. He has just started new roles as Director, State Agricultural Biotechnology Center and Director, Center for Crop and Food Innovations with Murdoch University. Prior to joining this university, Professor Varshney served ICAR as a Research Program Director of Accelerated Crop Improvement Program and Director of Center of Excellence in Genomics and Systems Biology. Professor Varshney is a globally recognized leader for his work on genome sequencing, genomics-assisted breeding, and translational genomics and capacity building in developing countries. He has made centrally important contributions towards improving food and nutritional security in India and Africa by developing key genomic resources in major orphan tropical crops such as PGNP, chickpea, groundnut, and pearl millet. His programs have delivered 11 superior crop varieties to some of the world's poorest farmers. Professor Varshney is a highly prolific author and a highly cited researcher. He is the youngest agricultural scientist and fourth Indian to achieve an H index of more than 100. He is the recipient of several noted awards, including the most coveted Science Award, Shanti Swarup Bhatnagar Award, and the most prestigious Agriculture Science Award, Rafi Ahmed Kidwai Award. Recently, ICAR won the 2021 Africa Food Prize for the outputs and impact of tropical legume projects led by Professor Varshney as principal investigator for 7 years. So we find ourselves extremely privileged to hear from you on the topic of genomic tools to address climatic change scenario to ensure food and nutrition security.
Yeah, Rajeev, so the floor is yours, sir. So
Thank you very much.
Yeah, so 45 minutes would be good enough for you, sir?
Yes, sir. Yes, sir. So even half, yeah, so that would
Thank
be
you.
good. Thank you very much. So hopefully that you can see my slides. Let me do it just a minute. Yes, sir. So you can see my slides.
Yeah. Go ahead, sir.
Okay, good. So before that, let me thank Dr. Vilas Tonapi, Director IIMR, for his kind invitation, and I'm thankful to IIMR and CAPA for inviting me for this talk. I saw many senior people, friends, collaborators in this meeting. So greetings to all of them from Perth. In India, you are having hot weather. In Perth, we are having very cold weather. So right now, 12 degrees Celsius temperature. So you can see I'm wearing that jacket. So this is because we are in the southern hemisphere. Anyway, and also greetings to everyone. And I was very happy to see Dr. CLL Gowda also in this meeting. And I think many other senior colleagues and well-wishers. So thanks and greetings to everyone. So as I have moved to Perth, and here I am working for that two centers, Center for Crop and Food Innovation and the State Agricultural Biotechnology Center. So I would like to highlight briefly about our work, what we are doing. But let me see, sorry, yeah. So here in the Food Futures Institute, we have three centers: Center for Crop and Food Innovation, Center for Animal Production and Health. This is related to the animal and livestock thing. And the other one is Center for Sustainable Farming System. And out of these three centers, which are the part of the Food Futures Institute, I'm leading the Center for Crop and Food Innovation. Very briefly, what this center is about. So basically through this center capitalizes on new technology and investment in crop agriculture, provides both strength and depth to undertake research on major broadacre and horticultural crops to improve yield, quality, and enhance tolerance and protection from biological and environmental stresses. And this covers all various cutting-edge R&D technology in agri-biotechnology, agri-technology, and food technology. And what we do in this center, we work to keep farmers competitive in the world marketplace and undertakes research in crop genomics, bioinformatics, genetics, transgenics, gene editing, molecular biology and physiology, soil and water management, new agriculture technology, including remote sensing and robotics, and the developing areas of food technology. So we are having several leading research groups affiliated to this center. And if you would like to see the major themes of our center, so we are having basically 6 major themes, and our lead researchers, they are part of the different themes. They are delivering on the different aspects. And one of the key theme is crop production. So we enhance the crop productivity by using the modern genomics, biotechnology, and crop breeding tools. The other important area is crop quality, because not only the productivity for us here in Australia, quality is also very, very important. And the other area is crop protection and biosecurity. And here we are talking about this biotic stresses and also about this pathogen invasion in the country. So the biosecurity aspect. Fourth theme is related to food technology, which is more related to the horticulture crops, that how we can enhance the quality of the food through these crops. And also the another important area is innovative production systems. And lastly, about the pathways to impact, which is more dealing with this gene editing technology and also the policy issues, etc. So these are the different areas in our center. And the other center which I'm leading, and this is about the Western Australian State Agricultural Biotechnology Center. And this center is a collaborative university center for all different universities in Western Australia. So agricultural and veterinary biotechnology universities in Western Australia, and also the State Department of Agriculture. It provides platform technologies and the world-class equipment and facilities for R&D in agricultural biotechnology to researchers from all different universities, state governments, and industry department. And our vision is being eminent, internationally recognized center for research and development. And as I said, so we are having basically more technological platform. So that's what we are doing. And through my chair, I keep on working with international organizations so that how we can contribute in towards agriculture and food security. So now coming to the topic, and I was hearing some talks from time to time, and I think this conference or webinar is very timely and very topical subject. And now when we talk about this climate change and agriculture, food security, I think before that we can see that, and we all are aware that at present, there are 5 global challenges in the world at international level. And one of them is that feed the increasing world population. And you can see that, well, this number keeps on increasing. So we need to provide the calories as well as other nutrition, including the proteins, micronutrients, etc. We need to meet projected energy demands. We need to manage the greenhouse gases, because when we talk about agriculture, they're also important contributor to the climate change. So we need to see that how we can manage, and we need to adapt to the climate change and variability. And we also need to preserve the natural ecosystem and biodiversity. We were having very good talks a few minutes back from Dr. Prasad, that he presented very nicely, and maintain global security, and agriculture plays very important role. Now we have heard from the different researchers and policymakers. Even yesterday we had really good talks, many talks, good talks. And when we talk about the impact of the climate change, and then there are already documented stories and research studies, etc. And they say that by 2030, 9 out of the 10 major crops will experience reduced stagnant growth rates, and this includes those important crop, maize, rice, wheat, etc. So price will also be increased when this will be the impact. Now, not only at international level, even in India, there are several groups who have provided that documentation, that what kind of impact we will have on the Indian agriculture from the climate change. And then this based on these different studies, and this is one of few studies, or one of several of those studies, which show that wheat yield will be reduced, rain-fed rice yield will be going down, and not only that one, on the different commodity crops also we will have that impact. So this is that issue of the climate change. And now when we are talking about the food security, if you see the hunger map coming from the World Food Program of the 2021, this clearly shows that still at present, around 811 million people around the world, they're not getting sufficient food. And 1 in 10 people do not get enough to eat. The worst part is that 25,000 people, including more than 10,000 children, die from hunger and related causes every day kind of thing. And hunger is also on the rise in the Western Asia. So, and climate, and also there are lots of conflict right now. We know Russia and Ukraine war. So this is also making the impact. So I think there are a lot of issues related to the food security. And we also need to make the color of India towards the green color, because if you see these different scale, that these are the countries where we are having 15 to 24%, 25% population undernourished. We need to change this color. We also heard from Dr. Bhandari also very nice talk in a few minutes back. So I think we need to bring this color also changed color in the context of India, but also in many other countries around the world. So now, and so we were talking about food security, but now when you talk about the nutrition security, and worst part is that even the countries like India, even the urban population, we are having the issues of the protein deficiency. And not only that, many times we do not know that we are having the protein deficiency. So I think this is also a big problem. For getting the protein, we all know that protein we can get from the animal protein through the meat or so, or we can get the plant protein. So, and we need to have the protein sufficiency in our body. I don't care that from which source this is coming. But now if you would like to see from the environmental sustainability perspective, when you are talking from the greenhouse perspective, water footprints in the on this planet, then we need to see that probably we need to think more to get these proteins from the plants. And this is about that. And not only that, that recently we also have published one analysis together with several colleagues who are involved in the human health, including Dr. R. Hemlata from the National Institute of Nutrition. And here we have summarized that even the issue about the micronutrients like iron, or so, that what kind of issues in the country we are having from this anemia. And then this is really that area that government is working a lot, but still we need to do more. So we need to struggle, we need to overcome the problem of this burden of anemia. So there are a lot of issues. Now, all of us, different disciplines, either this is agriculture or medical science or different areas, they're trying to tackle all these issues. But being agricultural scientists, what we need to do, we need to think 2-3 different areas. One, how we can breed for our crops, which will be the climate change ready crops or climate, or they can adapt to the climate change. Second is that how we can start or we can enhance the crop productivity in terms of those anticipated climate change. Third is that, well, how we can enhance the nutrition content, either enhancing the protein content or iron and zinc content, so that we can address these issues. So I think these are the key areas where we as an agricultural scientist from the plant breeding perspective, we can play important role. In this regard, a few years back, we wrote this article and opinion article in the Current Opinion Plant Biology, where we discussed that how the genomics can deliver climate change ready crops. And we had some discussions that, well, when we have the germplasm, we need to use the genomics and phenotyping technology. We use to the modern breeding technology. Dr. Sadanand also mentioned very nicely. He gave very nice overview. In my presentation, I will be giving some examples. And I think that by integrating all these things, we can develop the climate resilient crops. Furthermore, we also had another article where we have provided that approach. And I think this was last year or so, the 5G approach. And I say that this is a 5G approach for the crop genetics and breeding. The first G, this 5G is not that mobile technology, but we compared or we gave that when telecommunications is going in the 5Gs, I think the crop breeding should also go towards the 5Gs. And 1G is the genome, that we need to have well-characterized genomes for any crop species. And nowadays, a lot of advances have happened. Germplasm needs to be characterized at the genomic and agronomic level. We need to have the genes identified and their functional characterization. And then we need to use them through the breeding. I call this approach genomic breeding. And nowadays, the another approach called gene editing. And this is good that India is moving in the positive direction, that we will have these technology, especially coming from the SDN1 and SDN2, not regulated at GM, which is really very good thing for the country. So I think that we are having several approaches, and this is the research framework. And what I'm going to do, that will highlight some of the examples, and many of these examples, they're coming from our research, what we have been doing at ICAR together with partners, several ICR institute, including IIPR, Directorate of Groundnut Research, Indian Institute of Millets Research, also All India Coordinated Research Project on Pearl Millet, and many other state agricultural universities in the country and international collaborators. So in this perspective, if you see from the protein perspective, then the three legume crops, they play a very important role: chickpea, pigeon pea, and groundnut. And these are grown by the smallholder farmers, rain-fed crops. So, and they are a very important source of the proteins. And the other group of the crops, and this is about the sorghum and millets, where IIMR is working, ICAR is working, and many other colleagues are working. And I think that this dryland area we are having in India, 68% of the dryland areas. And in those areas, I think that these millets, broadly millets, which include sorghum and millets, so they are important, gifted, nutrition-rich and climate-resilient crops. And they address two major challenges about which we are also having this webinar about the food and nutritional security. And so these are very important crops. And I think that because of the hard work and of Dr. Vilas Tonapi and other colleagues from IIMR, ICAR, and the leadership of Director General of ICAR, Dr. Mohapatra, and Ministry, I think this is really good that India is taking lead to have this 2023 as an international year of the millet. So I think that this will be enhancing further. We are all aware that if you compare these different cereal crops, then sorghum and pearl millet, they really lead other cereal crops in terms of those different micronutrients. So these are so important. So what we need to do, that we need to work, or we need to enhance these different contents further in these crops. What we have done, we need to do, we need to integrate the genomics and breeding and other allied disciplines together. And we need to think from the perspective that how we can enhance the disease resistance, how we can enhance the crop productivity, pest resistance. We need to develop the crops which are tolerant to the different environmental stresses in context of the climate change. We talked about nutrition. We need to think about the environmental sustainability. And in this context, I will share some of our experience related to the genomics. And I think that not only recently, and I'm taking you maybe that several years back, more than 17 years back, in 2005, when I used to work in Germany. At that time point, people used to talk about the marker-assisted selection, marker-assisted breeding, because they used to identify the markers using the breeding. But at that time point, genomics was relatively new. There were only the genome sequence available for rice at that time point in 2005, and before that Arabidopsis. But so at that time point, together with Professor Andreas Graner and Professor Mark Sorrells, we gave a concept that how genomics can be used in the breeding program, and we gave this concept of the genomics assisted breeding that was published in the 10th anniversary issue of Trends in Plant Science, and the title was Feeding the World Plant Biotechnology Milestone. And after completing about, and then during this last 15 years, a lot of genomes came, a lot of genomics assisted breeding approaches were used. And 15 years later or so, then the journal asked, and they were having another special issue, 25th anniversary issue, Feeding the World, the Future of the Plant Breeding. And then in that one, they asked that, can we summarize that what happened during last 15 years in the genomics assisted breeding? Point number one. Point number two, where we need to go further, what kind of new approaches now we are having. And in this article, we described those results, and we also gave a concept, we call it GAB, Genomics Assisted Breeding 0.2. So basically that we are going in the next version or so. And here I will touch some of these points at later stage, that in the current scenario, how we can moving further. Furthermore, and just before this talk, there were the discussion, and Dr. Sadanand also mentioned, and Dr. Varaprasad mentioned that when we are talking about the crop improvement, we need to bring the diversity from the wild species from the land races. And recently we have given this concept, and then this was published as a cover article also in the Trends in Biotechnology, that how we can bring those important alleles which were lost during the domestication in the plant breeding process, and we can bring those alleles back to the elite lines. We can enhance the genetic base, we can reduce, or we can address the issue of this narrow genetic diversity in many crops, including the pulses or so. So this is also important, that how we can reap the crop wild relatives for breeding the future crops. So I think these are the premises, and now what we need to do, how we can do it. And for doing these things, as I mentioned earlier, I think nowadays there are lots of advances in the genomics technology, sequencing technology. And not only that, even the phenotyping technology, they have been advanced a lot. And right now with that new technological advances, including these precision phenotyping technology like X-ray, CT imaging, and different kind of methods, you can do very high precise phenotyping for the large scale crops or so. The other thing is that in the area of sequencing and genotyping, lots of advances have already have happened. You can develop the reference genome. Now we can sequence hundreds to the thousands of the line, and we can develop the pangenomes. I will talk about it later. And when you are having this sequencing, genotyping technology and phenotyping technology, you can map those any traits. And then not only that, you identify the genes, you identify the better version of those genes. We can call them haplotypes. So basically we identify the superior haplotypes, and these can be integrated in the crop improvement program. So this is the way that breeding needs to go. In this context, from the perspective of genomes for our crops, when I was working at ICAR-Indian, what we did that we led the genome sequencing consortium for many crops, and in several other crops we co-led with other international partners. As a result, we delivered the genomes of pigeon pea, chickpea, pulse millet. Also in the case of groundnut or peanut, two different ancestral species, A genome, B genome species. Then also cultivated species of two different kind of subspecies, Fastigiata and Hypogaea of peanuts. So we have published all these things. Everything is in public domain. And not only that, we also work with many other organizations and deliver the genome sequences of sesame, mung bean, azuki bean, jatropha, and just now this pea genome paper has also been accepted in Nature Genetics 2 days back. So we also have accomplished this important job, and then many other things. So what I'm trying to say, so now this is possible to decode the genomes in very fast manner and very precise manner in the less cost. And we had this privilege to decode or to deliver the genome sequence for more than 10 different crop species. Now, once you have this genome of one individual or accessions available, many people think that job is completed. And I say, no, job is not completed. Rather, your job is started now, because what happens when you sequence one individual, you can identify the genes of only that particular cultivar. And nowadays, there are the studies that if you sequence the other cultivar, cultivar 2, cultivar 3, then you realize that many genes which are specific to cultivar 2, they are not cataloged when you sequence just cultivar 1, or you don't find those genes for the cultivar 3 in the cultivar 1 and cultivar 2. So nowadays, what is happening that we are sequencing large number of individuals, and based on the sequencing of these different individuals for a given species, then you can catalog the genes for all genes for that particular species. So for instance, in the case of chickpea, we are sequencing one individual, we may not have all the genes. So we need to sequence larger number of individuals. This is called pangenome. So when you are having all genes of one species together, cataloging of those things, we call pangenome. We also extended this approach called super pangenome. And what we say, like even in the case of chickpea, for instance, and we are having 8 different species in Cicer genus, like so Cicer aritinum is the cultivated one, Cicer reticulatum, echinospurmum, Yamashita. There are many other subspecies or so. So what we are saying that not only for one, we are developing the pangenomes for each of those subspecies. And when you develop those pangenomes for all these different species, then at the larger scale, at the genus level, you can develop a pangenome, and we call it super pangenome. And when I was at ICAR-Indian, we have already worked in some of these directions. I would like to highlight a few areas of research in context of the climate change and nutrition security, that how the germplasm sequencing efforts have been helpful. In the case of pigeon pea, a few years back, we sequenced 300 pigeon pea lines coming from the 23 different countries. And this study provided a lot of important things, including the center of origin of pigeon pea here in Madhya Pradesh. But most important, from the plant breeding perspective, provided the genes associated plant breeding related traits in the crop species where we did not have anything. Similarly, in the case of chickpea, where we sequenced 430 chickpea lines, and we had the phenotyping data for yield under drought and heat stress conditions coming from ICAR-Indian colleagues and from many partners. and we identify several of those genes in that aspect. And not only that one, that when we have done the phenotyping of this reference set of 300 lines for many nutritional traits, because in this talk, I'm talking about the nutritional security as well. And you can see generally the pulses, they are having 20% protein content, but now some of these lines we got about even up to 24% protein content. So if you can identify those genes or so, and then we can ask a question, can we introgress these things in the elite cultivar so we can enhance the protein contents without that sacrificing the yield, because yield is the paramount. And not only that one, you can see many iron, folate, and many other micronutrients, large number of variation for this germplasm. And we have done some GWAS analysis for the nutritional traits. And for instance, for the protein content, we found several genes on the chromosome 8, chromosome 2, and chromosome 3, etc. So I think that this is really important to identify those genes. Similarly, we are also identifying the genes for different micronutrients. Some of my colleagues at ICAR, like Rakesh Srivastava, he has been working with different collaborators and colleagues, and he has also mapped some of those traits like iron and zinc for in the case of pulp millet in the different chromosomes or so. So I think that you can map those related traits or so. And this is coming from the smaller set. But now we have moved a larger set of the germplasm, and many of you may have seen this paper last year in Nature. And what we did that we have sequenced 3,366 chickpea lines, and this was a big one of those biggest plant genome sequencing study in any crop species where we have sequenced at the whole genome level, more than 20 to 30x coverage kind of thing. And this was covered in the New York Times, in the Economist, in many other important newspapers and journal articles. That this was showing that, highlighting that how important this study is and related to the crop improvement or so. And I will not go in the details, but would like to mention that out of these 3,366 genomes, we had cultivated, wild, and many other things, and we have developed the pangenome. So as I was saying earlier, so for instance, earlier from our chickpea genome sequence paper, which was in 2013, we identified about 29,000 genes. But when we have done the sequencing of large number of individuals, we found about 1,600 new genes which were not present in earlier genome. And many of these genes, they were found enriched in the different metabolic pathways, which are really important, associated with the adaptation for the climate change related trait, or in some cases, maybe with related to the disease resistance or so. And once you got those genomic information, and we would like to see that how we can use them in the plant breeding application, and plant breeder would like to see their genotyping cost minimal so that they can screen any line with the minimum cost. And many of you may be aware about the high throughput genotyping project, which I was leading at ICAR together with several other organizations. And through that project, we have brought the cost down for these 10 SNP panels, and where we can screen any line for about $1.5 to $2 per sample. But for each of our species, we have developed a range of marker genotyping platforms, starting with SSR, Casper, genotyping by sequencing, high density genotyping array or so. Our purpose was to provide a range of marker genotyping platform for genetics and plant breeding community, and depending on their objective, they can use any of these platforms. So for instance, recently we accomplished this 2,000 SNPs array, and they are being used now for the gene selection purpose or so, and I would talk about it. So we can do a lot of work in the genomics area, but second important thing is the phenotyping technology. And at international level, there are different advances in those phenotyping technology, where you can do the aerial phenotyping by using the drones, these helicopters, different kind of aerial vehicle, etc. And also you can do the lot of ground phenotyping platform, even under the roots, etc. So you can do a lot of work. At ICAR, when I was there, so together with our crop physiologist and pathologist and our breeding colleagues, we have developed different platforms related to drought, including Legiscan. Yana Kolova is leading these efforts, Lysimeter, and also for the field together through our plant breeders in different areas. They are really having the good platform. So what we did that for different germplasm, we have done the phenotyping by using these platforms. We use these approaches called linkage and association mapping approaches, and we have identified combining all these approaches during last many years, we have done mapping for 20 to 50 traits in chickpea, groundnut, pigeon pea, and this is the result of my several colleagues from Center of Excellence in Genomics and Systems Biology, and also our collaborators from ICR centers, State Agriculture University, and others. And similarly, in the cases of the cereal crops, pulse millet, sorghum, finger millet, including like Rakesh Srivastava, that and other colleagues, they have done a lot of good work and mapped those traits, 10 to 20 traits in the different crops. And now the next important thing is, so okay, you would develop the genomic resources, you understood the genes. Now the next thing is how you are able to deliver these things in the form of the superior varieties, because in morning also there were discussions, and also in that when we were listening about this talk from that ISRO, that now the design thinking, so we need to think that how we should be able to deploy these things. Sometimes we are having ideation, proof of concept, but they are not going in the deployment technology, which is very, very important. So what we have done, that in the case of genomic breeding, I would like to classify these approaches in genomic assisted breeding, marker assisted selection, marker assisted back crossing, marker assisted recurrent selection. Many times these approaches have been used. Now genomics assisted breeding 2.0, and here we are having the new approaches like haplotype-based breeding, genomic selection, also the gene editing. And gene editing, we are telling in two different flavors. Sometimes you would like to improve those genes, you would like to have the good allele, then we are calling this promotion of the allele through gene editing, we call it page. And sometimes you want to remove the bad alleles, we call it removal of alleles through the gene editing. So I think these are the new approaches we need to do. I'm very happy to mention that through collaborative efforts of our breeders from my former institute, ICAR-Indian, and also our collaborators in India, in Ethiopia, in Kenya, and many other countries, we have been able to deliver several improved varieties coming through genomics assisted breeding. So those information, genomes, this is not just restricted to those Nature paper or high impact high impact factor papers. They have come to the plant breeding level. They have these fruits of genomics assisted breeding have been delivered to the smallholder farmers in several countries, including one of these interesting example. And this was the first example where Ethiopian Institute of Agriculture Research, and together with us, we have delivered this variety called Jelly 2, and this was the variety which is having 19% higher yield under the rain-fed condition. In India, our colleagues like Dr. Bhardwaj from Indian Institute of Agriculture Research, also Dr. Shailesh Tripathi from IRI, and many other colleagues from Indian Institute of Pulses Research, also Dr. Mannur and others from State Agriculture University from Karnataka. So we have delivered the varieties for drought tolerance, for the fusion wilt resistance, and they are coming from the genomics assisted breeding. And also in the 2021, there are another 3 varieties came, and they were coming from that IRI, from Dr. Shailesh Tripathi and from Dr. K.R. Soren, and also from Dr. Aditya Garg. So all these people, they have been working all together with ICAR and all of us, and through the genomics assisted breeding, these varieties have been delivered. And not only this one, and just last week I heard from Dr. Bhardwaj that one of those lines coming from the genomics assisted breeding has also been released in Uttar Pradesh now through the State Variety Release Committee. So I think a lot of varieties, they're moving now from all this work what we did a few years back. And I have been told that several of these lines, they're already under the pipeline under this ACRIP trials or so. And similar is the case in the case of pigeon pea, where with the US Raichur, PJT SAU in Hyderabad, we have been able to deliver several of those lines. And in the case of peanut, where we started these efforts long back about this foliar disease resistance, though there were different issues during last few years, but this year, some of those lines have been identified for release. And I hope that in the next ACRIP meeting, this release thing will also be completed in the case of peanut for the foliar disease resistance. And some of these lines, they have showed really good expression of the resistance in Ghana and Nigeria, etc., which we developed in India also. And not only that one for foliar disease resistance, and through some of our collaborative efforts from Dr. Ramesh Bhatt, and he has delivered 2 lines and coming from the genomics assisted breeding and for the foliar disease resistance. And you can see Dr. Dalwai also when he was visiting this field few years back when I was there, and at that time the then Agriculture Minister of Karnataka was also there. So what I'm trying to say, so you can deliver these varieties through genomics assisted breeding. And not only for that diseases or abiotic stresses, but even for nutrition related traits. So for instance, if you see the normal peanut varieties you are having, if you see the oil composition, then you are having linoleic acid and also that linoleic acid, palmitic acid, and oleic acid. Linoleic, so basically so that there are the different kind of these things, and linoleic acid is not good for the health. So what we need to do, we need to change this kind of composition, and what we have done through genomics assisted breeding, we have developed the lines which are having more than 80% oleic acid, and through Directorate of Groundnut Research or so, the first set of the groundnut varieties, Grinar 4 and Grinar 5 were released, which were also included in the package when Honorable Prime Minister Modi, when he delivered this in the 75th anniversary of FAO, when he dedicated several of these varieties. So I think this is really good. Now, what we were doing further, that we were combining those foliar disease resistance and high oleic acid traits together. And some of these lines, I hope that they will be coming in that Acri pipeline, etc. So I am very hopeful about that one. Now, what we need to do further. So these were some stories from genomics assisted breeding from the first concept. Now we are moving to the GAV 2.0. What we need to do, as I said, so now there is the approaches haplotype based breeding, because based on the sequencing of large number of individuals, we can identify the haplotypes. Now we need to bring those superior haplotypes in the varieties. Genomic prediction is another area that now we do not need to screen the line for one or two traits. We can understand, we can screen the line at the whole genome level. We can predict the phenotype of the line at the whole genome level. Third area is optimal contribution selection, because sometimes when you bring the germplasm material in the breeding, many times we have the issue of the linkage drag. But now through this approach, we can do the selection of parental lines in such a manner which will deliver the higher genetic gain and minimum linkage drag or so. Genome editing is another approach. And now we believe that by combining a new approach, or like called speed breeding, where you can generate or where you can advance several generations in 1 year or so, then you can reduce this time also. So if you combine all these things together, you can develop the elite varieties, improved varieties in the context of the climate change or so. Now, very quickly, because I'm a big fan of this haplotype based breeding, what is this haplotype based breeding is that when you are sequencing larger number of individuals. So for instance, here you sequence individual 1, 2, 3, etc. And then you are having, based on the sequencing data for each gene, you can identify, for instance, in gene 1, which haplotype is present in genotype 1, H1. On the other hand, haplotype 3 is present for gene 1 in the genotype 2, haplotype 4 is present like this one. Similarly, you can do this haplotype mining for different genes across the whole entire germplasm. And then after that, you basically, what we do, that we do the phenotyping of these lines, and based on the haplopheno analysis, we try to identify that which haplotype is the good haplotype for the given gene. So for instance, in the gene 1, for leaf area, haplotype has 3 is better. For gene 2, for the root depth, haplotype H1 is better. For gene 3, haplotype H2 is better for the flowering time, etc. So you identify those superior haplotypes. Then you ask a question that your elite varieties, or the varieties which farmers are growing, what kind of haplotypes you have? And we identify sometimes, oh, for gene 1, this variety is having H2 haplotype. On the other hand, the best haplotype is H3. Same thing for gene 3, haplotype H4 is this variety, but we need to have the H2 haplotype. So now we need to change those haplotypes, and we can do this thing through haplotype based breeding. I think this is a really interesting approach. We have demonstrated the power of this approach in the case of our 3000 chickpea genome sequencing project, where these 3000 chickpea lines were phenotyped with our collaborative partners from at Sihor and also in ICAR-Indian, in Junagadh Agriculture University, in IIPR Kanpur, at 6 different locations for 2 different years. And then we combined all these things, data together, and then we have identified that basically haplotypes, and we targeted in the first instance about 67 genes, and we identified about 350 haplotypes, and we did a lot of analysis. I will not go in the details because of shortage of time, and we validated our approach. With the released 129 chickpea varieties, and we identified that some of those varieties which are good varieties, they're containing good haplotypes. And when you go further, then you are basically accumulating the superior haplotype, etc. And eventually we identified 56 promising lines that are having the superior haplotypes. and which are still not present in the elite varieties, and these can be integrated in those best varieties. And here I was also very excited to see that, and many of you may be aware, when Honorable Prime Minister was visiting ICAR on 75th anniversary, and then we had a pleasure that he visited the chickpea field where we had several of these lines, and it is so pleasing that when a Prime Minister is basically visiting the field where we are having these things and testing some of those things. So I think that this is a really interesting thing, that haplotype-based breeding, and this is the area that where we need to go further in different crop. Same thing for micronutrient analysis we have done across the 3,000 chickpeas. We have done also the, and then you can see the larger number of variation, and we can identify the haplotypes from the nutritional perspective. When you do the protein content analysis, we have found at least 25 lines which are having more than 30% protein content, and now this can be also integrated together. Lastly, I would also like to mention about this genomic prediction approach, a genomic selection approach. And when I was at ICAR, we have taken these studies and we have demonstrated that even the legume crops, and earlier basically cereal crops are always advanced, but in these cases we have shown that we can do the genomic prediction in chickpea, groundnut, and pigeon pea. And in the case of pigeon pea, we also provided the parental combinations for delivering the higher yield, which and they can give much more better hybrids also. So I think that this is that all these approaches are very, very powerful approaches. And very quickly, I will also like to mention in the sorghum and millets, which are a very important crop from the nutrition perspective. We know that one, and I think that India has done great work in both of these crops from the perspective of that cultivars. More than 150 cultivars released. Figures may be wrong. Probably IIMR is having much more updated data, but I think that if you combine with the seed industry, number goes is 300. But what I want to tell, that seed industry also has played very important role to promote this sorghum and millet cultivation in the country. And nowadays, not only that one, but an important thing is that about the biofortification, both pulse millet and sorghum is moving further. And I know some of these efforts in the case of pulse millet, even this again picture where we had some finger millet and little millet varieties, they were also released or dedicated to the nation by Honorable Prime Minister. And I think pulse millet is also the crop where ICAR has put a bar that when any variety or hybrid which is released, then we should have minimum number of iron and zinc. And I'm getting this information from Dr. Tara Satyavati, where they have demonstrated that during last 4 or 5 years, they have delivered several varieties or hybrids, several hybrids basically that, and then all of these things, they are having really high level of iron and zinc, and there are larger number of these things. So I think that this is really good. Now, next thing is either you develop hybrids or varieties through genomics assisted breeding or conventional breeding or biofortification method or by gene editing technology. The most important thing is that we need to have high yielding climate change ready crops with the better nutrition and with better profitability to the farmers. But now the next thing is we need to ensure that they are reaching to the farmers in the real time. And in India, things have been much more better situation. And I think, again, thanks to the ICAR, state agriculture universities and the policy makers, that we have done a great work in terms of the replacement of the old varieties by the new varieties or the hybrids. But there are still several countries in the world where we still have the issue. So what we need to do, and we have provided this approach, or we did some analysis, and we said that even when you are having these elite varieties from any method, we need to ensure, we need to have the rapid delivery system. And if in some countries these are already available, then we need to strengthen. If not, then we need to establish. And what does it mean? That when we are having these elite varieties, we need to strengthen the seed system, either through formal or informal way. And Dr. Tonapi knows much more better than anyone else we know about the seed system. And then what we need to do, that when they are reaching to the farmers, then farmers also need to provide it the better agronomic packages, because you can enhance or you can realize the full potential of genetics or genomes of breeding only if you are having the good agronomy packages. If not, then you will not be able to harness the full potential. And then for that, we need to have the digital decision support tools, and we can call this area digital agriculture, etc. So farmers need to be given all those information. The next thing is that we also lose a lot of yield during the post-harvest losses, and there are a lot of issues I think even yesterday when I was listening, Dr. Dalwai also, that they were talking that even we talk about the cold storage we see in the country, and more higher percentage this goes only for the potatoes. So now we need to see from all these perspectives that how these things can be addressed so that we should have the reduction in the post-harvest losses. And the next thing is that the farmer community needs to be connected to the markets, and so that we can have the minimal middleman or so, and farmers can ensure higher profitability. And also the value addition is very important, that how we can do this thing, value addition. So not only the farmers, even the consumers can be the beneficiary. So we don't need to think now just for enhancing the crop productivity, we need to think from the crop profitability. And I can tell you that though I have been here in Australia for last 3-4 months, here we are always talking that how the growers, the different thing is here in India and Australia is, which is thing that here that farmers, they call themselves growers, and large scale land, this is not a smallholder. So the first thing always comes that how this crop even enhance the crop productivity, will be enhancing the profitability to the growers or not. And all of our research are dictated by those needs. So I think this is really very, very important for us. Here, I would like to highlight an example of the tropical legumes project, which was initially led and conceptualized by Dr. C.L.L. Gowda. And then at later stage for about 7-6 years or so, I was leading this project. And this project is a successful example of integrating this plant breeding, genetics, seed system, capacity building, all these things together. And this was a big initiative for about 12-13 years from the Bill and Melinda Gates Foundation, which was having the partners ICAR-Indian, SIAD, IITA, and 13 different countries in Africa, 2 countries in Asia, India and Bangladesh. And we were having 6 different crops: chickpea, cowpea, groundnut, common wheat, sorghum, pigeon pea, etc. Massive efforts for large number of colleagues or so. This project delivered facilitated development and release of 266 improved varieties, facilitated production of 500,000 ton seeds, which worth about 6.1, so this could produce about 6.1 million ton grains. And all these success stories, they have been documented in the different formats, either in the special issue of the journals, books, etc. And I think that this is the way that we need to do the things. And I think that based on all these efforts or so, this project or ICAR was awarded that Africa Food Prize for the efforts or the outputs of these projects. So I think we are very happy. And this project has given, or the outcome, this thing is given several outputs or so. One is we need to have the long-term studies. We need to bring all these different disciplines together. And I think we always need to see from the pragmatic manner or so. Anyway, friends, now I think I would like to summarize my talk, and we have talked about this agricultural role of agriculture for food and nutrition security. In this direction, we believe that in general all crops, but millets and pulses, they play a very important role for nutrition, and because they possess the climate resilience. And if you are talking other crops, we need to integrate those traits there, and genomics and breeding innovations, they're key for accelerating crop improvement for both agronomy and nutritional traits. Nowadays, the fast forward breeding, including genomic prediction approaches, haplotype-based breeding approaches, they can expeditiously create and incorporate superior genes and haplotypes and crop improvement. I did not discuss this area, but I think this is the upcoming area about the systems biology, machine learning, artificial intelligence. And I think that nowadays we are working, even at Murdoch University, we are moving in this direction, that how we can integrate these approaches for enhancing the precision and efficiency in the crop improvement. Lastly, there is a continuous need for R&D investment from international, national, and local government agencies in both upstream science as well as translational research, because many times we would need to show the impact and we leave the focus on the upstream science, which is also not good. We should not do this thing on the cost of one of the other, but we need to have really good balance in research and development. So I think that's what I would like to say. And with these words, thank you very much for your patience. And I would like to thank all my previous collaborators and partners around the world. And thank you very much. I will be very